set a good example and stay on time. So we were a little bit late on the last session. I apologize for that. So I'm going to be responsible, and Antonio will make sure that I stick to 15 minutes here. But welcome to our third session, and really this one is going to be focusing on the next step. So we've talked about what are we trying to do with tracking data, what are the observation systems, and the current needs in the observation systems, and gathering the data. Now we want to talk about what we do with it. So how do we store it, how do we share it, how do we process it, how do we turn it into the types of insights that we need. And we'll be hearing from myself, Jill Deppy, Ben Carlson, and Sarah Davidson remotely today. So without further ado, I'll be talking about individual ecology at scale and bridging what I'm sort of calling the informatics gap. We have made the point about the burgeoning data streams that we have inbound in ecology several times this year. I'm going to make it one more time and just try to set the table of how astounding this is. So what you're looking at here on the left is maybe the first extrinsic animal marker ever. So this is a white stork from the 1820s, so roughly 200 years ago, that departed Germany for a location that was at the time unknown to European scientists. Right? We didn't know, nobody knew where they migrated, at least the Europeans did. This bird takes off and heads down to what we now know as Africa and survives barely a failed hunting attempt and returns to Germany somehow with this thing through its neck. And by locating the type of spear or arrow that is, they can get a really rough geolocation. And the astounding thing is this happened like 20-something times, right? Enough that they named it. It's called a file short, and they happened in those museums. There's 27 of them, mostly from the 1820s. We fast forward, this is a picture of my favorite and also objectively the best bird, flame-legged owls, carrying one of the migratory connectivity projects, pinpoint archival tag to sub 10 meter resolution. We're able to do incredible things. And then, of course, we've heard all this morning about these even newer technologies with even cooler properties, right? So Icarus, promising to reduce tag size and latency and spatial precision. And then, while not spaceborne, we missed not to mention this just a few weeks ago. The cover of Science was a tiny radio tracker on a moth tracking course-correcting behavior on a migratory flight, which is just absolutely mind-blowing. And of course, we've seen this graph a few times, and I'm guessing that Sarah's going to show us something similar. The amount of data that we have is growing at least exponentially. I think Martin said earlier something like 3 or 4 million records a day. At the time that these were made, there's something like 3.5 billion records on MoveBank. It's probably upwards of 4 billion now. And of course, that creates a bunch of challenges and opportunities, and there's lots of ways to sort of create a structure to it, and this is a very hasty attempt at thinking about this. So once we have these data, we need to figure out how to store and organize them. Where do we keep these data that are collected? How do we make those accessible for sharing and discovery? In other words, how do we maximize the utility of the data that we have or that we're going to have? Today, I mostly want to focus actually though on analysis. And what I mean by analysis is not just methods, but also questions. How do we turn these major data streams into ecological insights, either for basic research or for actionable management decisions on the ground? And I'm going to do that with 2 case studies, really one case study and then a very small one on the end, but an analysis that is trying to take advantage of some really big moving ecology data. And that's this. So what you're looking at here, I'll tell you how these data are collected in a minute, is human mobility over 2020 measured with cell phones. And one of the key features of this that you might guess is this right here, which is the period of lockdowns associated with COVID-19. It's a dramatic reduction in human activity on the landscape. So dramatic that a team of researchers led by Christian Roots and the International Biologging Society published a paper coining this the anthropause. And amid the tragedy of all of this, it actually does offer us an incredible opportunity to try to disentangle the mechanistic drivers by which humans modify animal behavior and the relationships with the environment. And specifically, it allows us to think about decoupling these things that are traditionally confounded, like human mobility, human activity, and our stuff. All of a sudden, our cities got quiet, and in some areas, our rural areas, our parks got very, very busy. And so this International Biologging Society actually started an entire initiative to gather a really large data set to facilitate a series of sub-projects in order to ask relevant questions about this stuff. It ended up having a team of over 600 data collaborators, including 13,000 plus tagged animals of over 200 species globally, and more than 1 billion location records, right? And that's just in the animal data that we have associated with just a couple of years here. Our team here at Yale, co-led with Reeve Oliver from UCSB and Diego Alasoto, a PhD student here in our group, is really leveraging these high-resolution human mobility data, and the United States in particular, because we can get high-resolution human mobility data thanks to our lax privacy laws, to try to disentangle these drivers. This is true. To try to disentangle these drivers. And our premise, again, I'm not going to go into all the details of this study because it's not quite about the study. Hopefully some of you got to check out Diego's poster, and I think you may hear some more about this from Ruth tomorrow, but really to talk about how do we deal with data of this size. But our basic premise was that we can calculate geographic space use on a weekly scale for all of these animals, and also their environmental niche breadth. So essentially trying to understand the variety of environmental experiences these animals are having as they move through space over time. And a very simplified modeling framework here where we're going to try to disentangle these. We actually fit quite a few very specific models here, but just for simplicity's sake. And one of the really cool things is that human mobility data that I showed you before. It's using a product called SafeGraph, working with one of our collaborators, Song Gao at the University of Wisconsin. And again, it's hourly cell phone data at a census block group level. So you can think about the spatial resolution of like a neighborhood. It actually varies across the United States. Pretty high resolution human data. And so just to walk you through kind of the idea in our workflow here, we're going to look at one cougar to start. So this is from Chris Wilmer's data set. They published a paper on this just outside of Santa Cruz in 2021. And what you're looking at here is one individual cougar. The darker points are 2020, so during the period of lockdown. And even just visually, you can see that in 2020, it's moving closer to Santa Cruz and moving into a higher, more human-modified environment. On the right are the individual points for that cougar annotated with this human mobility data. And it's pretty apparent. You can see in 2020, it's experiencing an environment with far less human activity. Of course, we can scale this up to do inference at the species level, and that's what this is doing here. And again, I'm not going to walk through all the fine-scale results, but you can see that human mobility dramatically reduces the animal space use. Human modification does, human mobility does. There's this interesting interaction effect where they actually moderate each other, and there's interesting reasons why that might be. And in some cases here, the human mobility is actually moderating the environmental niche breadth experienced by this animal. This is one relatively small component of our overall data set, and I told you that what we wanted to talk about, how do we do these things at scale to come up with synthetic insights. And so our sub-project involves roughly 12 million GPS locations, sort of after cleaning, so after we've gotten rid of outliers and irrelevant points, of 4,000 individuals and up to 42 species. Depends on what analysis it goes in pretty decent geographic coverage, right? Not perfect, but pretty large. Which is a large data set, much larger than I'd ever worked with, and I think much larger than most traditional movement ecology works with. And it takes a series of tools to be able to do that computationally efficiently. And here we really relied on the work of Ben Carlson, a PhD candidate, who's developed a series of really cool tools that allow us to do this in a scalable way. So the core of this is MoseyDB, which is essentially a relational SQL database that matches the moving data format roughly. It allows us to build these things and do these analyses without putting everything into memory at the same time, which is critical with these really large data sets. He's coupled this with a series of tools that also make it easier to do the types of analyses that we need to do. So MoseyGet actually allows us to build this out of MoveBank from the command line. So we can actually just request the studies from a coded interface and build this database that then sits on our local machines. MoseyEnd is an annotation tool that allows us to annotate these data to anything that is on Google Earth Engine, which is incredibly powerful because it's not only taking advantage of everything that's on Google Earth Engine, but you can also think about customized layers that you could create in Google Earth Engine for really bespoke purposes. MoseyView is an R-Shiny app basis for really data exploration, visualization. And then Breezy is then sort of, I think you would call it a philosophy more than a rule set. It's a philosophy for reproducible workflows. This has a lot of really neat features. Here, the key thing is that it's easily parallelizable, right? So that when we're trying to do these operations over thousands of individuals, we can do so in a really efficient way on high-performance computing platforms. Which is important because what we ended up doing was developing something like 80,000 weekly space use estimates across these individual animals using dynamic Brownian bridge models. We also use an analytically tractable way to do this for environmental niche breadth. I'm not going to show you results for today. But what you get is something like this, right? Where we can actually understand the relative contribution of modification and mobility and whether there's an interaction between those factors across a whole suite of species. We're working with this data to try to develop a set of typologies that would also describe this as more of a generalizable framework. So like how do we think about which types of animals respond in which ways to humans? When you condense this down, it turns out there's actually only 3 or 4 ways that animals are responding. I don't want to go too far into the results that are just too detailed for the study, but it's too cool to show. We can also show that this isn't just environmental filtering. When we take the animals where we have paired observations, we actually see a change. If they experienced a change in the human environment from 2020 to 2019, it also modified the species within an individual, right? So it's a behaviorally plastic response, which I think is very cool. Right, so what do we do with that? So that's one paper where we built a set of computational tools and workflows that allow us to turn this into ecological insights. But obviously there's more than this single paper. So associated or sort of in parallel with this project, we developed this website, animallives.org. And you heard Walter talk about this quite a bit on Monday. I'm not going to walk you through all the features. In a way, it's sort of a platform in search of a problem or in search of trying to narrow in on the problem space. But the idea is that it's a bit of a playground where we can deploy these computational tools to develop synthetic insights at scale. So right now it's just using a few individuals from a few species from the original Icarus experimental data. But we've taken advantage of those workflows that we built for the COVID project to be able to estimate species using dynamic boundary bridge models and autocorrelated kernel density estimates, and then of course pairing it with pre-annotated measures of the environment. QR code if you can go to it. But one of the challenges, so I just want to give you one example of the type of challenges we haven't solved that should be fundamental, really. And that's a track segmentation. And so one of the things that we found early on in this process, so we tried to use autocorrelated kernel density estimates, which of course have a range residency requirement. And they're actually very finicky about this. If you catch just a few migratory points in your range residency, your estimates of species absolutely explode. It looks like you're using space over something the size of an entire continent. And that's okay if you have a few animals or a system that you know quite well. You can go in and hand segment it. And in fact, with Animal Lives and some other projects, that's what we've done. This is a really cool shiny app developed by Fabiola Angarilli, who's a postdoc here with us that allows us to go in and actually click on these transition points and segment the tracks by hand. It works incredibly well, and it works well with our relational database structure even. The problem is we can do this for a few dozen species. We can probably even do it for a few hundred species. But if we're talking now increasingly about data sets that are hundreds or thousands of animals, it's simply not a scalable solution. It's just not how we can move into the future if we're trying to develop these new types of insights. To that point, and more broadly, I would advocate that we need tools which are scalable, flexible, and efficient. By that I mean tools that we can use for these new types of animal analyses that are global in scale or large geographic scale. They're flexible in the sense we can deploy them across multiple taxa, multiple environmental situations, and that they're computationally efficient, right? We can't rely simply on an army of research assistants that are hand segmenting tens of thousands, right? Think about billions of tracks in GroupBank growing at an almost doubling rate annually. And then I would also argue that we need the questions to match, right? The big data revolution from movement ecology can't simply be that we're doing a better job of the same analyses at finer resolution. There's an opportunity here to integrate these analyses and ask both new questions, where we're taking a comparative approach and a multi-species approach, a multi-context approach, and also I would advocate old questions. I think there are old questions that we have not been addressing with individual scale data simply because it's not available. And I think that when we see this burgeoning data set, there's actually some of these questions we might be able to tackle in a better way if we can build the tools to do so. And so I think I've remained on time, and Antonio can tell me whether I have, but thanks to all of our data providers and everybody that's helped with the project. Did it.